Hello and welcome to my video where I'll be teaching the biological concept of turning DNA to protein. Let's start with the basic, like what exactly is a cell? A cell is the smallest unit of living things. But isn't an atom smaller? The answer is yes. Atom makes up molecules and therefore molecules make up cells. This is why it's important to emphasize the living. Now you'll never see a cell like you see in your textbook, so you can compare it to a city. All the functions you'd find in a city you can also find in a cell, like the city hall, factories, territory, and border patrols. In this video I'll be drawing the animal cell. The cell membrane, which is permeable, would be the border patrol. It allows certain things in and blocks others out. Right now I'm drawing the lysosomes, which would be the junkyard of the city because it breaks down all the waste in the cell. The ribosomes are the factories that produce protein. This is very important because we'll be going back to this in the video. Then we have the mitochondria, which are the electricity companies that give power to the city, the same way that electricity companies give power to the city. Gives power to the cell. In the center, we have the nucleus. This is like the city hall and the library because it holds one of the most important information, the DNA. And around it is the nucleus cell membrane. Then we have the ERs. The smooth ER creates fats and lipids. The rough ER packages and delivers them. So these are factories and distributors. Finally, we have the Golgi complex or the Golgi apparatus. This is also a factory that packages and exports. Now let's move on to the actual process. The process of DNA to protein is a two-step process. Transcription and translation. Step 1, transcription. This takes place in the nucleus, and it's the process of transcribing information from the DNA to the mRNA, which stands for messenger ribonucleic acid. But before we begin, let's look at the DNA. As you may already know, the DNA is a twirling double helix, so it appears to be like this. But if we were to straighten it, it would simply look like a ladder. Despite what you think, you may think that the strands are identical, but really they're different. The first is 3 to 5, the second is 5 to 3. And in the center, the steps are the nucleic bases. Now let's take a zoomed in look at the bases. In this video, I present them like a jigsaw puzzle for a very important reason. Here we have 4 types of nucleic bases: thymine and cytosine, which are the pyrimidines, and adenine and guanine, which are the primines. A and T can only go together, and G and C can only go together. They can't fit in any other way. Which is exactly why I put them in a jigsaw puzzle position. They can't fit with any other except for the ones that they're assigned to. A to T, G to C, C to G, T to A. And it can go no other way. Now, for the transcription process, if we were to look from afar, it can be compared to a zipper with two zips. The first zip would be the enzyme RNA polymerase, which would be the promoter. Its job is to unwind the DNA. Then the RNA, which is a single strand, would come in and attach to a single strand of the DNA and transcribe the information. They'll continue on like this until the end of the DNA. The last polymerase has the job of rewinding the DNA. And that would be the end of the transcription process. Now if we look up close at the nucleic acids, this is basically what's happening. They move aside, and RNA comes in and fills it up with the corresponding components, except U is replaced instead of T, U for uracil, instead of thymine on the RNA. And they're rewinded and put back together. Now you may wonder, well, which strand is transcribed? Now if we go back, you can see that the strands are not the same as I said before. It is strand 3 to 5, that is the template strand, or the uncodable strand. This is the strand that RNA attaches to and transcribes the information from. And therefore, it is identical to 5 and 3. This one's called the coded strand. Now, you have to understand that not all the DNA is actually transcribed. The beginning is not transcribed because it's the promotion. This is the start signal. The middle is transcribed, and the end is not. We've officially gone through DNA to mRNA. The transcription process is over. Step 2. Translation. You leave the nucleus, enter the cytoplasm, and use the ribosome to help. Now you have to understand that 3 of these equal 1 codon, which represent 1 amino acid. Amino acids are the building blocks of protein. We'll use the red dots to connect them, making a peptide bond. tRNA, which stands for transitional RNA, has the anticodons on one side and amino acids on the other. So, with your mRNA, we go to the ribosomes, which have 3 stations, A, P, E. A stands for amino acid, P stands for preparatory, and E stands for exit. So the tRNA, comes into each station, puts in the anticodons, drops off the amino acid, and exits. Here's what it looks like. It puts in all the corresponding pieces with U and attaches an amino acid. As time goes on, they all attach, and then you have protein. The translation process is officially over. You now have protein. Thank you for watching, and I hope you enjoyed this video and learned a lot.